Hello everyone. Good afternoon. Welcome to the third Philippine Planktology lecture series. This is the first lecture out of the three lectures on the third year of the Philippine Planktology lecture series. Planktology lectures started during the pandemic, and all of the lectures during that time up until now are given through online platforms. And after 3 years, we are still here communicating and spreading science with you guys. The Philippine Planktology Lecture Series is not possible without the Institute of Marine Fisheries and Oceanology, or IMFO, the Philippine Planktology, the Philippine Society, the Plankton Society of the Philippines, and the Plankton Niche. Today, we are graced with 149 participants, and we have 57 participants via Zoom, and we also have participants via FB Live. So our participants came from the Philippines and Taiwan and composed of students, university and college, state university and colleges, non-government organization, national government agencies, the local government units, researchers, retired government employee, and other private sectors. Aside from Zoom, as what I said, we are also live via FB. The lecture will also be posted in the info page. So this is our program, and I will be giving you our house rules. So participants are requested to turn off their cameras and keep their microphones on mute. And also, please hold your question until the end of the presentation, and question will be entertained thereafter. To ask question during the open forum, kindly raise your hand and wait for the moderator to call you in. You may also type in your question in the Q&A box. Please do not spam the chat box. Kindly accomplish the evaluation form later on. So without further ado, I will be presenting to you our first speaker for this Plankton's lecture series. So she is a lecturer of the Department of Biological Science, National University of Singapore. She is a biology lecturer at the said university in the Department of Biology and Sciences and has more than 10 years of research experience in phytoplankton and zooplankton research. She worked on the ecology of several cyanobacterial taxa, phytoplankton and zooplankton interaction, microplastic impacts and eutrophication, and algal bloom mitigation in Singapore's reservoir and coastal environment. She teaches various biology and ecology courses for undergraduate students and several courses for master's program in the said institution on biodiversity, conservation, and sustainability. Help me welcome our lecturer, our speaker for today, Dr. Maxine Mowe. Ma'am?
Hi, thank you, Jency. Thank you very much for that kind introduction. That was really, really nice. I'm so happy to be here. I'm really honored that I was invited by the committee and by the colleagues here. Let me just share my screen and get started. Okay, can you see my screen?
Yes.
Okay, great. I think without further ado, I'm gonna get started so that we can carry on. And then please feel free to keep all your questions and you can ask them at the end of the talk. Even if it's something outside of the field of what I'm talking about, please ask me because I love to answer questions. Okay, so I think I want to just give a brief intro about myself before I get started. So my name is Maxine. I've been in the Department of Biological Sciences since 2010. I started my PhD here, and then I went on to do a postdoc looking at phytoplankton. In my PhD, I was really focused on cyanobacteria. In my postdoc, I started to look more about how plants interact with cyanobacteria and can help suppress cyanobacterial blooms. And then now I'm a lecturer within the department, so I teach a lot about this, but a lot about other pollutants as well. But I wanted to focus my study on what I love best, which is phytoplankton, of course. I'm going to be talking about some of the work I've done in the past few years in Singapore, but also some of the more wider studies within Southeast Asia, Asia, and also in the Philippines, and what's currently being done. And then we can talk about what to look for in the future and what gaps are we still trying to fill. So without further ado, this is my email address that I'm showing you in the side here. So if you want to email me next time, go ahead. And this is also my Twitter handle. So sorry, let me just Okay, so this is the overview for my talk today. I'm going to give a background on what kind of blooms I'm talking about, what is cyanobacteria in general, and then I'll talk about some of the research I've done in Singapore in the past few years. I'll talk about some case studies in Southeast Asia, mainly focusing on the Philippines, and then like going forward, what should we study in the future and why? Why is it important? So let's get started. So when I first started to think about cyanobacteria, I first started to even consider doing cyanobacteria for my own PhD work. I was really actually more interested in phytoplankton and zooplankton interactions and not just strictly phytoplankton. But what happened is that cyanobacteria came and really just overwhelmed and took over my entire thesis because there's so much that was not done at that time and there's so much that we didn't know about cyanobacteria within Singapore. So just very briefly, cyanobacteria are prokaryotic organisms. They are photosynthetic and they are found in almost all environments on Earth, so freshwater, marine, and terrestrial habitats as well. The reason why there's been more interest to study them in the recent years is because they can form harmful blooms that can be toxin producing. So, what exactly are these blooms? That's what I'm going to go into next. So, how we define an algal bloom is basically that they are high, it consists of a very high cell count within any water body. It can be a river, it can be a lake, we're talking about up to 10 million cells per mil of just one or two taxa that are dominating the phytoplankton community. These taxa can be cyanobacteria, but they can also be other types of phytoplankton that do not produce toxins. We can also use a definition where we take the chlorophyll A concentrations, and if the chlorophyll A concentrations are above 20 microgram per liter, we consider that a bloom environment. The WHO, or the World Health Organization, actually have drinking water guidelines for microcystins, which is one of the toxins that cyanobacteria produce, and it's currently 1 microgram per liter. So as long as the drinking water is below this concentration, the water is safe to drink. But if it's above this concentration, then it is not exactly safe to drink. And I'll talk a little bit more about that in the coming slides. So these are just some photos that I've taken over the years of different water bodies in various places. And you can see I'm holding like a packet here. It's not a green wheatgrass drink. It's actually a water sample taken from a water body. So when the cyanobacteria or when phytoplankton is very dense within an environment, you will get water that looks like this, that looks entirely green, and it floats on the surface of the reservoir or the lake, as you can see in this first picture right here. And it is looks a bit like spilt green paint. So that's how you identify cyanobacteria from other phytoplankton because they have gas vacuoles that allow them to float on the water surface. So when we're thinking about cyanobacterial blooms, there's usually three very main important variables that determine whether a bloom will develop in a particular area. So that is light, temperature, and nutrients. And I'm going to talk a little bit more about these three factors and how they affect cyanobacteria growth and toxin production in the coming slides as well. But I wanted to give you a little bit of an intro, a little bit of an understanding of what these blooms look like in the field. So like in case you go out to the field, and I know a lot of you are undergraduate researchers and you are doing your own sampling, you can take note of these different blooms that are happening in different areas. So let me just start this video. So this is kind of a very typical cyanobacterial bloom. So as I said earlier, a lot of the phytoplankton or the cyanobacteria is on the surface of the water body, and it has sometimes if there's disturbances in the water column, there'll be gaps as you can see here, and a big patches of green here on the other side. So let me go to the next slide. So these species, not all species of cyanobacteria produce toxins, but those that do, they can produce a variety of toxins. So they can produce hepatotoxins, neurotoxins, dermatoxins, cytotoxins, and lipopolysaccharides. There's many, many different kinds of toxins. There's also many theories behind toxin production, like why do cyanobacteria actually produce toxins in the first place? What is behind their toxin production? So some of the theories that have been put forth is that they are defending themselves from herbivory, that it's a byproduct of other cellular functions. So it's just produced as an output or as a byproduct. Some studies have said that it is an allelochemical to prevent growth of other phytoplankton and macrophytes. And there are many, many other theories. And the reason why we call it theories is because there's not one agreed upon understanding of why they produce these toxins. We only know that they produce them in different amounts under different environmental conditions. So a little bit more about hepatotoxins, because that's what I'm going to be talking a lot about in this lecture. The main hepatotoxin I'm going to be talking about is actually microcystin, which is the structure is featured here. It is the most frequently occurring. It affects liver function and can lead to death within hours. So, of course, if you are getting affected by hepatotoxins, it will be either through direct drinking, or sometimes what has happened in the past in some of the case studies or some of the impacts, it's caused by direct contact with the blood and contaminated water sources. For example, dialysis, liver dialysis, or kidney dialysis. So any of these processes that have direct contact between the blood and contaminated water bodies. So we have microcystins, which are named after Microcystis aeruginosa, which because it was first isolated from this particular cyanobacteria. There are currently 85 different variants that are recognized. And the toxicity of each variant is different. And the most toxic variant is actually Microcystis LR. So no need to worry too much about the LD50. It just means that it's the mean lethal dose, or it's how much is needed to kill 50% of the population. So the most toxic will be about 50 microgram per kg, whereas some can be in very high concentrations and they are not toxic at all to people and to livestock. So some of the impacts of cyanobacteria blooms, besides having the toxins that lead to fish kills and lower oxygen concentrations in water bodies, they can also reduce light to other phytoplankton and macrophytes, because as you saw that photo I showed you earlier, there was a layer on the surface of the reservoir, and that kind of prevents light from entering. So it's very hard for other submerged macrophytes or other phytoplankton to grow in that kind of environment. There's also reduced energy transfer to higher trophic levels, because cyanobacteria tends to be quite unpalatable to higher trophic levels. And then, of course, reduced fitness of macrophytes because they can't get enough light. So they eventually die off due to light limitation. So some other impacts of cyanobacteria blooms, as found in other studies, would be high costs. Basically, you have to find alternative water supplies, and then you have to actually treat the water after you extract the water. So don't worry too much about this. But in Australia, it costs approximately about $240 million per year to deal with cyanobacteria blooms, because it contaminates a lot of the potable water sources. So they are a country that actually struggles yearly with cyanobacteria blooms, especially in the period that they are most affected, which is December to January every year. So I'm going to move on now to talk about phytoplankton blooms in Singapore. So phytoplankton blooms or cyanobacteria blooms in Singapore have been recorded for more than 50 years, and it's because we have a very highly urbanized catchment area for of our 17 reservoirs, which are man-made. So 2/3 of our land area is actually catchment for these reservoirs. So far, the highest toxin concentration has been found in one particular reservoir, Kranji Reservoir, and it was about 2.66 microgram per liter, which, if you remember, is actually higher than the WHO allowed limit. And more than 50% of our reservoirs actually experience algal blooms on a regular basis. So this is kind of what the blooms look like. Even in 2012, we had blooms. In 2020, we are still continuing to have blooms. Um, There's also this program in Singapore called the Active Beautiful Clean program, which has been instituted by the Public Utilities Board, which is Singapore's national water agency. So they are trying to encourage the citizens to take more active ownership and to clean up the waterways. But this also adds into the fact that if we want people to interact more with the waterways, for example, through water sports, through recreational activities, we have to make sure that the water is clean enough for them to come into contact with it. So another very important part for us is because our surface water is one of Singapore's national taps. So we don't have natural lakes. So we have to depend on these 17 reservoirs as part of one of our national resources to get clean water. So water security is a big issue for many countries, and Singapore is not above this threat. And we are constantly in negotiations with other countries and also trying our best to be self-sufficient in terms of our water sources. So we rely on desalination, new water, which is very very strong and reconstituted sewage water, but basically it's been filtered, ultra filtered. And we have been struggling with this for many years, and it's something that we will continue to face as we look at climate change and we have more needs for clean water in Singapore. So far in the past few years, we have had a lot of toxicology and ecological studies in Singapore cyanobacteria since 2015 to 2020. Basically, a team of researchers, including myself, Prof. Darren Yeo, Prof. Simon Mitrovic, and various other graduate students and undergraduates have been working on this type of work for years. And so we've hopefully had tried to add a little bit more to the understanding of cyanobacteria, especially within the tropical area and within Southeast Asia. So coming back to my diagram again about looking at what factors are really important for cyanobacterial blooms, throughout the work that we did in the past few years from 2015 or from 2010 when I started my PhD until now, we looked at nutrients, temperature, and light separately to try and figure out what were the impacts of these different conditions on cyanobacteria growth and toxin production in laboratory experiments, but using strains that were isolated from Singapore's reservoirs. So very briefly, I don't want to go too much detail about the experimental methods, but we did a nutrient experiment where we enhanced the nutrients within that those bottles to try and see what was the impact on the cyanobacteria, and we used these 3 strains of Microcystis. So for the growth effects, we found that actually nitrogen was more important than phosphorus, and it increased the yield of all 3 Microcystis strains. So this is not something that was unusual. This is something that we kind of already knew. We just wanted to make sure that we had a good understanding of how nitrogen is affecting these strains in Singapore. Then we went on to look at how nitrogen and phosphorus affect toxin production rates. So what we found was actually quite interesting. When we were increasing the phosphorus for all 3 strains, we actually had a decreasing amount of toxin production. So this was also seen in other studies, but not so clearly shown as how we did it here within this particular study. So this was very neatly done, as you can see. As we increase the phosphorus, as you can see these bars over here, we actually decrease the toxin production over time. And for the studies, some of the speed strains were more obvious than others. Yeah, especially for these two, for this particular strain, which is Microcystis ictioblabi, versus the other 2 strains from two different reservoirs. So then we looked at temperature because we wanted to see how does temperature actually affect these strains, and does the growth and the toxin production actually change with different temperature treatments. So we exposed the temperature to expose the Microcystis to 4 different temperatures: 27, 30, 33, and 36 degrees Celsius. Currently in Singapore, our water bodies kind of experience 27 to 30 degrees Celsius. Some water bodies tend to be a bit hotter, so they will go up to 31 to 32 degrees Celsius, but we haven't really hit 33 degrees Celsius, and 36 degrees Celsius is more of a climate change scenario where that's really, really warm water. So we had also a 28 day experiment, and then we wanted to see how the growth was affected. So what we found for these 5 strains that we carried out these experiments on was that the growth rates were higher around 33 degrees Celsius, and they tended to fall when the temperature was increased further to 36 degrees Celsius, which is not uncommon. So we can see here that the optimal temperature was around 33, and then for one of the species it was around 30 degrees Celsius, whereas for the rest they were similar for all the temperatures. That means they're able to grow well under a range of temperatures. Looking at the toxin production for under different temperature regimes, we found that the toxin cell quotas were actually higher around 30 to 33 degrees Celsius. But once you increase the temperature further all the way to 36, you can see that the toxin production actually went all the way down for each cell. So this is in a way good news, meaning that if we increase the temperature all the way to 36, we're going to get lower toxin production. However, if our temperatures only increase to 33 degrees Celsius, we're still going to get some increase in toxin production. And this kind of trend was visible for 3 of the strains, but 2 of the strains had decreasing toxin production as we increased temperature. So this actually indicated to us that all the 5 different strains had different effects or different reactions to different temperature regimes. So then we came to look at the light um, in the reservoirs, and we tried to look at different light environments to see how that affected the cell growth and the toxin production as well. So we carried out basically these experiments under 4 light intensities, so 10, 30, 55, and 120 micromole. So given that these light intensities are actually very low compared to what is experienced in the field, but this would be a typical light scenario for a very turbid reservoir or very turbid water body. So it's not uncommon to get quite low light intensities, especially if there's high turbidity in reservoirs. So we saw here that we had for the Microcystis ichthyoblabi strains, we had higher growth rates at low compared to high light, whereas for the Aeruginosa strain, we had lower growth rates at low and medium compared to high light. So we had 2 opposing effects of light. So as I said earlier, all of the strains react differently to the different environmental conditions. And there was no significant difference for the last 2 strains actually, which is Flos aqua and Viridis. So looking at the toxin production, you can see here that there were no significant difference for Macrocystis ichthyoblabi and Aeruginosa, whereas for Viridis, it had a decreasing trend as we increased light. For the other 2 strains, we actually had an increasing trend with increasing light intensity. So there's many different factors that are playing within this realm. So it has to be actually researched a little bit further to see in future what is really going to happen overall within the water bodies in Singapore. So overall, what can we learn from these laboratory experiments? I'm sure I've inundated you with so much information and your mind is just like, oh my goodness, there's so many terms, so much microcystis cell quotas, basically it just means how much toxin is produced per cell. So what we can basically learn for Singapore was that the current nutrient conditions and the low ambient light was actually very conducive for microcystis growth in Singapore, and lower phosphorus actually leads to higher toxin production. We also found that the high toxin production for some microcystis species in Singapore is a bit worrying because these species are common within the reservoir, and if they have really high toxin concentration or toxin production per cell, what does this mean when they have a bloom? How high is the toxin production going to get? So we also, after we did these experiments, we also had a lot of other questions, like how do we regulate blooms and growth of cyanobacteria? How do we deal with the dominance of different cyanobacteria taxa that are happening within the reservoir at different times? So there's many, many factors that still are currently under research. Another takeaway was that we found that for Singapore, climate warming may not affect growth as much, but it will increase toxin production at least until 33 degrees Celsius for most of the microcystis isolates tested. And because of the very different toxin production for different species, we do need like a species level ID for microcystis, especially when we're doing water quality monitoring. Because as I said, the different species had such different toxin cell quotas that it was really difficult to kind of come up with one nice, clear picture about what's happening with different environmental factors. And then there's also a lot of lack of previous studies of microcystin cell quotas for other microcystis species besides aeruginosa. So in the literature currently, microcystis aeruginosa is highly studied, but the rest of the species I've talked about today are not very well covered. So this is what I was talking about when I meant the very different toxin production. So you can see here that you can see that for Microcystis ictioblebbi, the maximum microcystin cell quota is about 1.3 femtogram per cell, whereas for aeruginosa, the maximum was up to 196 femtogram per cell. So, so the number of cells that would cause it to go over the drinking water guideline. So for Microcystis ictioblebbi, it had to be a bloom condition, like 1 million cells per mil. But you see, for species like Microcystis aeruginosa, you only need 7,000 cells. And that's it. You have gone over the WHO drinking water guideline. So this is pretty worrying because it kind of shows you that there's different number of different number of cells that are needed for each of the different species. So can we look at them all together as one genera? And I think the answer is no. We have to look at them as individual species. Okay, so I'm gonna move on to phytoplankton case studies in Southeast Asia and talking a little bit more about how are temperate versus tropical blooms different. So I have a little kind of a diagram for you here, just to do a compare and contrast between temperate and tropical blooms. So temperate blooms, so this is a picture of Lake Erie on the left here. Lake Erie is a very famous, one of the famous Great Lakes in the US, and they have very bad blooms, as you can see. They can even witness their blooms from satellite imagery. That's how big the bloom is. But they usually occur during the summer months, and they're very predictable in the fact that they occur within this June to August to September period, and they last for short periods of time within this timing, because then autumn comes around, the temperatures drop, and a lot of the microcystis kind of goes away until the following year. Whereas for the tropics, as you can see from this map I've shown you here with all the red dots, that's where I have actually denoted the cyanobacterial blooms within the tropics. They can occur throughout the year and they last longer. They can last up to a few months. And what happens is that when one cyanobacteria kind of dies down, another cyanobacteria takes over. So we're getting constant cyanobacterial blooms, but it's just led by different taxa within the phytoplankton community. So looking at tropical Asian cyanobacterial blooms, because I wanted to look at really Asia for this talk, based on a review I did in 2015. So what I'm showing you here is a graph, and the red circles are microcystis. Where microcystis was found, the red stars are where microcystins were found. That means the toxins were actually tested, and then so on and so forth for the other toxins that were found there, either spermopsin or other toxins. So majority of the blooms in tropical Asia were actually caused by microcystis, especially Microcystis aeruginosa, which is usually the prime target, the prime culprit. 66% of microcystis blooms actually produced microcystins. That means they had the toxins that were tested. And blooms in Myanmar and Indonesia were up to 2015 were not really tested for toxins. That they were noticed that the blooms were there, but they were not really tested for toxins. And these papers really lacked a lot of taxonomic detail and toxin analysis. So it's likely that they were underestimates of actual occurrences. There were also microcystis blooms that were denoted in Bangladesh, Sri Lanka, Vietnam, Thailand, Singapore, and the Philippines, and they actually were recorded to have toxin levels above drinking water guideline, which is above 1 microgram per liter. So this is just looking at all of the tropics and what the percentage of the different occurrences of different cyanobacteria taxa. So you can see that in blue here, most of the blooms were caused by microcystis, except in Australia where they were caused by Cylindrospermopsis. And that's no different than when we compare to Asia to the rest of like tropical Africa, tropical America. For the prevalent cyanobacterial toxins as well, most of them were found to have microcystins, and Asia is no different from Africa and America. So we can see that here. And when we did some analysis to look at, okay, which were the main factors that were actually driving these blooms. So we found that maximum temperatures were leading to more cylindral, or were being correlated to more cylindrospermopsis blooms, whereas higher total nitrogen was found to be linked to microcystis and anabina blooms. And this is very really ties in with the work I did earlier in Singapore, basically talking about how nitrogen is really important for microcystis bloom formation. We also try to look at how microcystins as a toxin is related to cell concentration in these blooms in the tropics. So we found that tropical Asia and America had a very strong positive relationship between microcystins, MC is short for microcystins, and cell concentration. So it's a very nice positive line here. Whereas for Africa, it was a very weak positive relationship between MC and cell concentration. So possibly there's other factors that are kind of playing into that. That's why the relationship is not as high. So I want to go into a very short case study about Laguna Lake, and I'm sure all of you are familiar with Laguna Lake. And forgive me if my information is a bit old, because this is based on quite a old paper that I found. I found a newer paper that was based on prawn fishing ponds. But I want to talk about Laguna Lake first, because I'm actually very interested in hopefully someone following up this work. And I hope someone in the audience here is working on phytoplankton. That would be great. So basically, Laguna Lake is one of the largest lakes in the Philippines, and it's tropical. It's located in Luzon, southeast of Manila. I don't need to tell you that. The surface area is about 922 square kilometers. It has about 100 rivers and streams that drain into the lake, and the outlets are Pasig River to Manila Bay. So the thing about Laguna Lake that makes it interesting is because it's so shallow. The average depth is 2.5 meters, and the watershed is agricultural, industrial, and mining. So it's also used for fisheries. So it gets periodic cyanobacterial blooms that affect fisheries yield and, of course, the drinking water in this area. So they recorded microcystis blooms. The highest was in 1996 so far. I'm not sure if they've done it follow-up studies since then. So later in the question and answer, maybe I'll ask the audience whether there's been more work that has been not been published yet. I'd be very interested to know about that, which is about 4,000 micrograms per gram. And there are about 9 variants of microcystins found, and MCLR was the most dominant. And remember what I said earlier about MCLR being the most toxic variant as well. It's still very understudied compared to other temperate regions and tropical regions as well. And we still don't know what is the impact on the flora and the fauna. So how were the fish impacted when these blooms occurred? Were there fish kills? Was there low oxygen because of these cyanobacteria? There's really no information about that. So I'm really, really interested in hopefully someone carrying out this information. That's why I wanted to include this case study. So yeah, another question I had was, what are the recent cyanobacterial toxin information, the concentrations? I managed to find this kind of article from ABS CBN News in June 2019, where they are still having cyanobacterial blooms within Laguna de Ibay, within Laguna Lake. So you can see this photo here. This is obviously cyanobacteria, looks like microcystis to me. So I'd be very interested to see whether anyone has been following up this work. The more recent case study that I just managed to find is about looking at prawn aquaculture ponds in central Luzon in Philippines. So this study was carried out to identify and characterize the phytoplankton, but more specifically cyanobacteria in these prawn aquaculture ponds. So they found that Oscillatoria, agardi, princeps, and Microcystis aeruginosa and Weisenbergi were the ones that were more commonly occurring, whereas Anabina was only observed in one site. So these were the different taxa that they found. These are all photos from their paper. So they found Anabina, Oscillatoria, and Microcystis. So they also stated that these are possible harmful algal bloom formers. That's what HAB stands for. And they can produce toxins. They can also form high biomass, which may affect the prawns, which may affect any of the other flora and fauna within those prawn aquaculture ponds. So they also kind of reflected on the fact that it could also be due to the poor water quality of the ponds, the poor monitoring and management within the farm, and the link between the nutrients and the blooms is still unknown. So there is still studies that need to be done to try to link how do the nutrients within the ponds themselves actually lead to these cyanobacterial blooms, and why do some cyanobacteria dominate and not others, and what's the current toxin production that's going on in that pond. We have no idea. So this is just putting it out there that there is some work done, but there is a lot of space for more work. So I'm sorry, I feel like I've talked for a very long time, but I'm still going to finish up just very briefly, just about a few more slides. I wanted to kind of cover what should we study in the future and why. And I'm going to cover it within a Singapore context, and then I'll cover it in the region as well after that. So what should we study and why? So I think it'll be very interesting to study the interactions between phytoplankton species, like basically the community level dynamics, to see which taxa are taking over and when and during which period of time, during monsoon, intermonsoon, and so on. I've also got some undergraduate studies looking at nutrient limitations in Singapore and how that can lead to better management of the catchment and reduction in blooms. So this study is ongoing. We're still slowly going through the phytoplankton data because we all know how long that takes to count. So this was carried out by one of my students, Candice, and it's also kind of being followed and continued on by another student of mine, Stanley. So far we found that of the 6 reservoirs, one was nitrogen limited, but the rest were mostly phosphorus limited. Some were coal limited, and that means that they were both nitrogen and phosphorus limited. So even though they are experiencing blooms, they are still limited in their nutrients. And how we manage each of these reservoirs would determine which strategy we take based on which nutrient is limiting. So that's why it's very important to know which nutrient is actually limiting when you try to manage reservoirs. So for the region itself, actually what we found was that our analysis and our meta-analysis and our correlation agree that on the general understanding of cyanobacteria that nitrogen is important and so is high temperature. But also another thing to highlight or to mention is that many of these water bodies without information were not included. So I couldn't include all of the water bodies in tropics. There are so many. I could not include all because many of them were not even sampled, or some were just anecdotal information which I could not include. There's also a possible bias because of the low frequency of sampling and the short sampling duration, because you need to sample for several years at a time. So some of the new information that we need would be the cyanobacterial patterns of occurrence, the toxin production, and the causes of the blooms. That means we need to link together the nutrients, interactions with the higher trophic levels to really understand the total ecosystem. So as I mentioned, some of the more recent cyanobacteria studies were done in Philippine prawn farms by Tabayan in 2018. Some Malaysian lakes also have looked at the presence of cyanobacteria, like that Senang did in 2015, and also in an Indonesian lake in 2019. But we're still lacking a lot of toxicological and ecological studies. So we don't know what factors are really affecting these cyanobacteria, what impacts does nutrients have, what impacts does light have, or temperature, what toxins are dominant, how do these blooms impact local livelihood in terms of the potable water, the drinking water, as also water to water their crops and their farms. How does this cyanobacteria blooms affect food security? That means looking at that link again between cyanobacteria blooms and the prawn aquaculture farming, or cyanobacteria blooms and using that water for agriculture, watering, and so on. So what are the conditions for high toxin production? Like what under what conditions do these blooms actually form? Like what conditions are we talking about? Are the conditions the same throughout Southeast Asia, or is it different in different areas based on rainfall, based on monsoon season, and so on? And then finally, what are the effects of climate? And with increasing climate change, we already know that there's more and more stress being put on our water resources. And a further stress is having to deal with increase in not just a decreasing amount of water in our water bodies. That means a higher evaporation rate. We're dealing with higher evaporation rate coupled with concentration of nutrients, and that leads to cyanobacteria blooms. So it's like a kind of a positive feedback loop where low water leads to higher amounts of nutrients in the water, and that leads to higher toxin blooms, and then it goes through that whole cycle again. So there's a lot of work to be done, and I hope people in the audience are interested, they are excited, and they want to study a little bit more about phytoplankton in general in the Philippines and in Taiwan. So thank you very much for giving me this time to talk about cyanobacteria, to talk about Singapore's work, to talk about the region. I want to thank the Third Philippine Planktology Lecture Series, Louis, and the rest of the organizing team. I want to thank the Department of Biological Sciences, where I'm working, and the Public Utilities Board that's paid and funded a lot of the work that we've done in the past. University of Technology Sydney, where I carried out some experiments, and all of our previous students who have contributed to this work. So I'd like to take any questions that the audience has.
Okay. Thank you so much for that wonderful presentation, Max. Yeah, I think a lot of our audience are really interested because, as we all know, like Philippines has like different big lakes, and then we're on a yearly basis we're experiencing like algal bloom, and then people will only be aware of that these things are happening when there are fish kills, like
Yes.
when the fishes are affected. So
Mm-hmm.
it will now start appearing in the news, and everyone will be informed what algal bloom is.
Yeah.
So we've consolidated questions from the chat box and also from the FB Live. And then we have a lot of undergrad students from the audience.
Mm-hmm.
First question, I think we'll go with the basic one, which is like, how does cyanobacteria look like? Do they form structures, for example, like chain-like aggregates, or only when they are blooms? So in general, I think like, do they form like colonies, or do they form like big aggregates during algal bloom?
Okay, so maybe can I just escape and then wait, let me just, because I think I have some photos. Sorry, I have to go back. Ah, here. So cyanobacteria actually can be in chain-like forms or filamentous. So like, for example, like oscillatoria or here, or it can be a chain form like Anabina, and it can also form colonies like the microcystis colony you see here. So they're very similar. So these were taken in Philippines, but actually the species that I'm finding in Singapore are also very similar. So the species in Singapore, sorry, let me go back all the way. The species in Singapore look quite similar as well. So, oh, sorry, let me just, yeah. So you can see my screen.
Yep.
Okay, so the species in Singapore look more like this. They have these basically they can form colonies, and the colonies, based on the colony shape, that's how we actually identify them to species. So for example, this colony up here on the left hand is actually Weisenbergi because of the very strong mucilaginous outline that you see. This colony is Flossaqua because of the shape of the colony. So usually when we are trying to identify to species, we will identify it from their colony formation. For the filamentous group, they can form basically also blooms where they kind of stick together, but they don't form a colony for the filaments. They just kind of lay on top of each other. Yep.
Mhm. And I think like a follow-up question from the undergrad students, it's related with the microcystis, like how hard it is to isolate and culture microcystis variants. Like I think they want like to know like what are the usual materials that you're using for culturing. What are
Okay. Yeah, no problem. Okay, so I use very low tech, very low tech instruments to culture them. So for example, if I go to the field, as I said earlier, and I collect this bag full of a highly highly dense bloom, what I'll do is I'll come back to the lab and I use a lot of glass pasture pipettes, a lot of 24 well plastic plates, and I do a serial dilution technique. So I pick up one or two colonies under the microscope and I basically just inoculate them into media. And the media which I use, I like to use MLA media, but you can use a variety of different media. It's just that it's important for you to make the media yourself so that you know exactly which nutrients you are putting into that media and you can tweak it according to what you want. So for me, I've had the most success with liquid media, with MLA media. But of course, you can also do, there's many different techniques. Serial dilution is one of them. I also use another technique where I basically pull pasture pipettes under a Bunsen flame, and I get really thin glass ends where I use that to really just pick one colony at a time. So I think the painstaking part is making that isolate grow and making it pure. That means one species alone. So that sometimes is the challenge. So it takes years to get basically cultures up and running. It took me, I think, easily 2 years, 2.5 to 3 years initially in the start of my PhD. And I was very fortunate to have a research assistant that was helping me as well, because I don't think I could have done it alone. Yeah, I was going out to sample all the reservoirs in Singapore, and it was just a lot of work. So I think that having good culture media is one, having sterile conditions. So every time I did any subculturing or anytime I did any proliferating or trying to continue the culture, I did it in a laminar flow. So I never worked outside due to contamination. And I grew all of the strains in a cabinet, like a closed cabinet system in those cell culture flask bottles that have a filter cap. Yeah.
It's a long time, like 2.5 years.
It is a long time, but some of these strains that I cultured or that we isolated in 2012 are still alive today. So they've been growing for 11 years. And I
So
can't bear to let them go. So I still keep them growing, and my students are still using them. So yeah.
once they are conditioned in the lab and then they're used to the conditions, they
Yeah.
will become like stable and easy to maintain.
Yes, then they become very easy to maintain, but they tend to lose their colony formation. So once they're in the lab and you're growing them for years, they don't really grow there. They don't really have their colony formation anymore, and they
Mhm.
grow as single cells.
Okay.
Yep.
Yeah. Yeah. And then going to like the Singapore scenario when it comes like
Mm-hmm.
to deal with algal bloom,
Mm-hmm.
do you have like does the government have like a remediation control program for algal bloom in different reservoirs in Singapore right
Um,
now?
yeah, currently what they've been doing is so that the project that I did for my postdoctoral research was part of that. It was to actually use plants as a natural kind of biofilter and a competitor against these cyanobacteria. And it worked very well in very small experimental setups. So like 100 liters to 1,000 liters, it worked very well. But the moment that you put plants into bigger areas, it tends to be harder to control the cyanobacteria. And you have to have a lot of plants to cover the entire reservoir. So then you start to have competing problems where you have, if your reservoir is full of plants, and then you have other problems with low oxygen and mosquito breeding. So there's always a fine line and a balance between these two. But they are exploring using plants, I think, to kind of control cyanobacteria in some reservoirs. And some reservoirs are actually quite clean and clear because of the fact that there are submerged plants in these reservoirs. And of course, the catchment area is mostly forested. A forested area or intact forest, which also leads to a lot of filtering of nutrients by plants that don't get filtered out into the water body. Another thing that they do is that they also do a lot of artificial destratification, which means that they have bubblers placed throughout the reservoirs to try and destabilize the water column. So once the water column is destabilized, especially for cyanobacteria, you can prevent their growth because cyanobacteria has gas vacuoles, whereas for other phytoplankton, they don't really mind this tossing and turning or this disruption. But for cyanobacteria, they don't grow as well if the water is not stagnant.
It's
Yeah.
really so one of our audience commented like in Laguna Lake,
Mm-hmm.
they are constantly observing abundant water hyacinth, like
Mm-hmm.
macro in the lake. So they are asking like how do you think like the abundant macrophytes affect dynamics in algal bloom? Like
Yeah, that's a really good question. Actually, I didn't really manage to cover it here, but actually, there is a competition between the phytoplankton and the macrophytes. And with water hyacinth, as you can see, I think I also put in a picture, and there was hyacinth in that picture as well. Sorry, let me just go to the next. Here, sorry, it's a bit slow. Uh, so for water hyacinth and for cyanobacteria, they do compete with each other, but a lot of times they have to have. So you can see here, there's also water hyacinth right here. There has to be a lot of water hyacinth in order for the competition to win between. In order for the competition between macrophytes and cyanobacteria to be won by macrophytes. Also, the another issue that comes about by using plants is that when the plants start to die, their nutrients just go back into the water column again, and then the cyanobacteria will continue to proliferate.
mhm,
So it really depends on what are the conditions of that prevailing reservoir, and if Laguna de Bay has already got a lot of water hyacinth and it's still having blooms, that means the nutrient concentration within the reservoir is really very high. So in order to actually control the cyanobacteria blooms that are happening within this lake, there has to be some control of the nutrient inputs coming into the lake. That means
mhm.
there has to be either stringent rules of the diffuse sources of nitrogen that are happening within the catchment area. Yeah.
Let's see. It's really interesting. And then Sir Lawrence Vito commented, I think it's from the limnological station in
Mm-hmm.
University of the Philippines in Los Baños. So there is still scarcity of studies on algal bloom in relation to fish kill. Right now, they are doing like phytoplankton monitoring and water quality monitoring in 7 lakes in
Mm-hmm.
Laguna. So that's his comment. Like there, like in the Philippines in general, there's still like a lot of like we're still lacking in terms of like experts and at the same time effort in monitoring
Yeah.
systems. But yeah, I think like monitoring water quality and then phytoplankton abundance could help us like have a sense of like early warning when it comes to
Yeah,
algal bloom.
definitely. I think the one of the better early warning system, like let's say if counting phytoplankton or counting microcystis is too tedious, is actually taking chlorophyll A samples. Chlorophyll A samples are very, very useful, and it's done very, very quickly. So that can just be done by a water sample that you collect, you filter onto a filter paper, you carry out extraction using ethanol, you run it through a spectrophotometer, and you'll immediately know what's the phytoplankton biomass you're dealing with on a daily basis or a monthly basis, or you can do it even every week because it's quite fast to do. But then again, when it comes to controlling that phytoplankton, that's another ball game altogether. That's something else totally different. But I understand that it is quite challenging to carry out this work.
Mhm,
And that's why I want to encourage more people to do it so that it gets done.
mhm,
And because I think there is some expertise happening within Philippines itself. It's just that how much of that work is really channeled to the natural lakes. Because I feel that um like so this looking at this Laguna Lake is that's why I'm more interested in the natural lakes in the Philippines. Looking at the prawn aquaculture ponds, that you're kind of expecting these prawn aquaculture ponds to have very high phytoplankton growth anyway, because they are feeding the prawns very high nutrient pellet or food. But for the lakes, that's more a bigger concern to me, from my opinion, because the lakes are what house all your endemic and your native species. So to me, actually, these lakes are more have a more pressing need to be researched, like to
oh,
really see how fast eutrophication is happening, how is eutrophication affecting the native endemic, like copepods, for example, how are they affecting the growth of these particular copepods. Yeah.
mhm. Yeah, and then we're actually running out of time. So I think we're down to the last question. Like
Okay.
we have social scientists in the audience, and then they're wondering like how the Singaporeans are perceiving like the concept of like algal bloom and how it's affecting like the way of living in general. Like do you have knowledge how people are willing to invest and to be part or to be involved like in controlling algal bloom in the reservoir?
I think currently a lot of people are not really aware
Mhm,
of certain blooms. It's only those people who are more, I would say, more in tune with what's happening, like more nature lovers. They go out to the reservoir more often. They will notice these things. But because like as I said, those reservoirs that are open to recreation, those reservoirs will obviously get more scrutiny than
mhm,
other reservoirs because, as I mentioned, not all reservoirs are actually open to recreation.
mhm.
So those reservoirs that are open to recreation, definitely people are aware of the
Yeah.
water quality and they do want to do their part to kind of keep the water clean. And already I think the government and the National Water Agency in Singapore does quite a lot to prevent nutrients from reaching the reservoir by kind of removing a lot of the farm area, changing the catchment area to try to prevent nutrients from entering the reservoirs. But of course sometimes it's very, very difficult because especially if the catchment is really urbanized, it's really, really difficult to prevent nutrients from entering the reservoirs. But people are aware, especially if it affects their recreation, for example, their canoeing, their kayaking, their contact with water bodies in Singapore. But mostly Singaporeans don't really do a lot of swimming in the reservoirs. They're mostly kayaking on the surface or they are fishing.
Yeah. So,
So because of the lack of swimming, I think that realization of algal blooms is not so big. It's not such a big impact to them, like, wow, the water is this color. How am I going to swim? Whereas in Europe and, like, for example, the Great Lakes,
mhm,
swimming is a normal thing in summertime. They all go down to the lake, they have picnics, they kind of make a big event out of it. And so when there are blooms at that time of the year, it's very, very devastating to them. Like they are really affected, the people, and then they will really investigate or they will force the government to do more in their control of cyanobacteria. Whereas that's not really a common practice here in Singapore. Like people will do picnics or have picnics by the reservoirs, but they're not going to enter the reservoirs to swim,
mhm,
for example, because that's really not allowed actually. Like it's not actually allowed. Like you can't really go inside the reservoir to swim. And the people who really enter it are those who are doing their canoeing training, their kayaking training. Yeah.
and I can attest to that. Like right now, like it's summer here in Canada, and then all
Yeah.
jumping to the lake because it's too hot for them.
Yeah. And I'm sure the lakes there are also experiencing blooms, right?
For sure. Yeah.
Yeah. Yeah.
We still have like a lot of questions from the audience, but I'm sure you gave your email address and then
Yes, I
also
did.
Twitter handler. So I think like they're free to email you if they have like specific questions about
Yes.
microcystis and like algal bloom.
Mm-hmm.
I'll proceed with the program and then like this will be a very short one. And so I'll pass this one to Jency.
I'm, I'm very, I know that a lot of you really eager to ask questions to Dr. Max, but you can always connect with her through whatever platforms you prefer. And
Jency, I think your mic is being blocked. So yep.
sorry, is it? Can you hear now? Okay. So again, thank you very much, Dr. Mark Louis Lopez, our discussant for today. And I know that everyone still have a lot of questions for Dr. Max, who gives really a good presentation. You can always connect with her through whatever platforms you prefer. Yes. And before we go to the awarding of a certificate to the speaker, I would like to thank our participants. We have 93 participants here in Zoom, and we have 29 participants via Facebook Live. So let's continue. So, certificate of appreciation is awarded to Dr. Maxine, Allayne, Darlene, Mowe in recognition for her outstanding contribution as keynote speaker of presentation titled Phytoplankton Studies in Singapore and the Region, What's Come is Still Plugging Us, at the third lecture series of the Philippine Planktology on July 12, 2023, signed Dr. Sheila May Santander de Leon, signed Dr. Christy Acabado, and signed Dr. Mark Louis Lopez. Thank you very much, Max.
No worries. Thank you so much for having me. I really enjoyed talking about cyanobacteria and phytoplankton. I'm gonna put my email address in the chat so you guys can write to me, or you can also contact me on Twitter. So my Twitter handle is Maxine Mowe, and it's usually I update about cyanobacteria and phytoplankton there. So you can also write to me there.
Yeah. So thank you very much, everyone. Um, Before anything else, I would like to plug the degree programs offered by our institute. We have Bachelor of Science in Fisheries. We also have Master's Program and PhD in Fisheries. And also, these are the requirements. You can visit our Facebook page, the Institute of Marine Fisheries and Oceanology. So thank you very much, everyone, for attending. And please don't forget to answer the evaluation form. And see you in our next lecture on July 17 at 2 p.m. again. Once again, thank you, Dr. Max. See you soon. See you in the future. And yeah, here is, let me post here. Okay.
Yeah, Maxine, thank you so much for your time. We really appreciate it.
Realize I'm muted. No worries. I really enjoyed talking about what I love most, which is phytoplankton. So I'll be happy to answer more questions if people have questions.
Okay, thank you so much. Like we'll keep in touch. Like on
Yes,
like we'll see
definitely.
like in the freshwater summit conference in the future.
Yes, if anyone wants to invite me to come to Philippines and to do sampling, hey, I'm
Or-
definitely open to that. Okay,
Thank you.
thanks. Bye
Bye.
bye.
Nancy, thank you.